Hi, my name is Rebecca Walker, and this is my eReport for Cell and Molecular Biology, Unit 2. In this eReport, I will be covering 8 topics, including cell membrane structure with the membrane proteins, transporters, and the energy requirements, the formation, docking, and fusion of clathrin-coated vesicles, the formation and structure of cytoskeletal fibers, cell crawling via actin, G-protein coupled receptors, including cyclic AMP and calcium as second messengers, the cell cycle, including the roles of CDKs and cyclins in its progression, cell junctions, all types, and lastly, cancer as a sociopathic cellular condition. So as mentioned, our first topic is cell membrane structure. So the cell membrane in most eukaryotic cells consists of what is known as the phospholipid bilayer. The purpose of this membrane is to separate the inside of the cell from its environment and allow for different things to be transported in and out. This layer blocks mostly the diffusion of most hydrophilic molecules. The structure of this bilayer is true to its name. There are 2 layers of lipids facing opposite directions. Lipids are water insoluble, meaning they can't dissolve in water. They have hydrophilic heads, which are the red circles on the edges of this bilayer shown here. These heads are polar and have charged groups. They also have 2 tails attached to them, which are nonpolar. The hydrophilic and hydrophobic ends of these membrane lipids make them amphipathic, which allows for the membrane to be formed without energy. The fact that these phospholipids are amphipathic also means that if there is a group of them placed into water, the hydrophobic tails would huddle together to avoid the water, while the hydrophilic heads would interact with the water. This forms a micelle, as we see in the top of this image. It's essentially one single layer of lipids formed into a ball. The difference in membrane lipids, as opposed to the ones in micelles, is that their hydrophobic tails are too bulky. Also, membrane lipids have a carbon-to-carbon double bond, creating an unsaturated fatty acid. This makes that tail kinked, and because of that feature, along with the bulk, the tails can't fit right into a micelle, and the lipids have a harder time packing together. To combat this bulkiness so a membrane can be formed, the phospholipids line up tail to tail. The heads face the water both on the inside and outside of the cell. This usually makes a hollow ball, called a liposome, but once you put items such as organelles inside that liposome, it becomes a cell. Because these membranes need to be finite sheets with no free edges, the membrane needs to roll itself into a self-sealing ball, and it is able to do that all thanks to its structure. Because hydrophobic regions need to consistently be away from water, they need to huddle with other hydrophobic things. The components can't break free from the membranes, but they can move side to side, forwards and backwards. This is known as lateral diffusion, and it's what makes this a 2-dimensional fluid. As seen in the top of the image, this only includes moving through one layer of the membrane, not both. The lipids can't regularly move upside down, although they do about once a month. The bottom part of the image shows that lipid moving to the bottom layer of the membrane from the top layer. This is known as transverse diffusion, though it must be reversed by flipases. This is because the lipids that are on the inner portion of the membrane differ in structure and in function than those on the outer portion. So if they didn't go back to their original positions, it would make the membrane homogeneous, when in fact it needs to be asymmetrical to function properly. The density of the membrane lipids is what allows them to be more or less fluid. There are often gaps in the membranes due to the kinked tails of the phospholipids. So if the membrane needs to be more dense, cholesterol fills those gaps and the membrane becomes less fluid. The orange figures in the bottom left and top right of this bilayer are those cholesterols. As we can see in the image, on the outer portion of the membrane, there are glycolipids and glycoproteins. The glycolipids are lipids with complex sugars, known as oligosaccharides, attached to their heads. Glycoproteins also have complex groups of sugars attached to their outer-facing portions. This allows there to be a sugar layer, also known as a carbohydrate layer. This allows the cell membrane to be more protected from the outside world. It also absorbs water, which makes the cell more lubricated to be able to fit through smaller areas. In terms of the different membrane portions, there are many. One of these is integral proteins, which we can see as a teal oval in the middle of the bottom layer of the membrane. These proteins have some or all of their amino acid chains embedded into the hydrophobic part of the bilayer. Integral proteins are known as transmembrane proteins if they go all the way through to the hydrophilic regions of either end.
So this integral protein on the right is considered a transmembrane protein. If there are proteins that aren't embedded in the membrane, they're known as peripheral proteins. They can either be covalently linked to a phospholipid head, as we see with the yellow oval on the inner portion of the membrane, or they can be non-covalently associated with another integral protein, as seen by the green pentagon. The lipids are mainly there for structure, while the proteins exist to transport molecules across the membrane or receive signals. A good portion of the functioning of these proteins relies on the conversion of energy and shape changes, such as the work of bacteriorhodopsin and retinol. Retinol is a non-protein molecule that's inside of the protein bacteriorhodopsin. When retinol absorbs photons of light, it provides enough energy for retinol to do a shape change, which makes bacteriorhodopsin change shape too. This shape change causes a proton to literally be squeezed out of the protein, and once that energy is used, retinol and bacteriorhodopsin return to their original shape to start the process over again. The cell membrane is actually quite fragile, so it also has a cell cortex underneath it to make sure it's stable. An example of this is the red blood cell cortex. In this image, we see the protein spectrin, which is a squiggly purple line. It connects the bilayer through the transmembrane proteins, and without it, the red blood cell would lose its shape, which would then cause it to lose its function. This shows that cell cortexes are vital to the function of that cell. A type of transporter in the cell membrane is a channel. These are proteins that have no moving parts, and because they basically just sit there, they don't require any energy. This is because they rely on ions following their concentration gradient. Channels are specific to their ions, meaning if the ion is not the right one, it's unable to pass through. There are different types of ion channels, and they work to keep the resting membrane potential the same. Some ion channels are always open, meaning the molecule can pass through at any moment based on its concentration gradient, which is facilitated diffusion. In other ion channels, however, there are specific gates that are there to allow the ions to pass through or keep them out. The first of these is known as the ligand-gated channel, which we see in the top left of this image. With this, another molecule must bind to the channel to open it. For example, in the image we see the channel is open when there is a neurotransmitter attached to it, but once that neurotransmitter is gone, the gate is closed. The second type of channel is stress-gated channels, or in this image, the mechanically gated channel. With this, any type of physical stress will open the channel. So in the image, we see that the pressure causes it to open and allow the ion through, but once the pressure isn't there, the gate is shut. The last type of channel is a voltage-gated ion channel, where the channel opening is blocked by a voltage sensor. It's sensitive to changes in the membrane potential, so when that changes, the voltage sensor moves to open or close the gate. Transporters are proteins with moving parts, and they're able to move molecules against their concentration gradient, from one side of the membrane to the other. They have to use shape changes in order to do this. The molecule that's trying to pass through needs to fit perfectly to the binding site on that transporter, and only then will that transporter change shape to force the molecule across the bilayer. This is more of a turnstile rather than the trapdoor of the channels. Because transporters move and change shape, they require energy. One type of transporter is the coupled transporter. These use molecules moving with their concentration gradient to power the movement of another molecule against its gradient. So as we see in the image, sodium is moving down its concentration gradient, while glucose is trying to move up its own. Glucose uses that opening of the transporter from the sodium to get the energy to go where it needs to go. Another type of a transporter is an ATP-driven pump. These use energy from the hydrolysis of ATP to force the movement of the molecule against its gradient. An example of this is a sodium-potassium pump. This is an ATP-dependent pump, so it's not only a protein but an enzyme as well. First, sodium binds to the pump, along with ATP. Then, ATP is hydrolyzed, which harnesses the energy needed to change shape and allow sodium inside. Sodium can then be released outside of the cell membrane, and because of that shape change, potassium can come in from outside the cell and bind. The phosphate that was transferred to the pump after ATP was hydrolyzed can then be released, which allows for the channel to go back to its normal shape, and potassium can be released into the cell. This process is considered active transport. At the cell membrane, each vesicle that's necessary for transportation of molecules starts off as a clathrin-coated pit. It's almost like a basket, and it has a clathrin mesh as its base. Adaptins are used to recruit and secure the clathrin to the vesicle.
The more clathrin that's added to the vesicle or pit, the more that it starts to take its correct shape. Once the vesicle is formed correctly, dynamine is recruited to form a ring around the neck of the vesicle. This ring constricts the neck until it's at its breaking point, and then allows the vesicle to be released from the membrane as a clathrin-coated vesicle. Also, in addition to recruiting and securing clathrin to the vesicle, adaptants also help to choose the cargo that will be put in that specific vesicle. There are transport signals on proteins that are meant for other targets, and these bind to cargo receptors which are vesicle membrane bound. Adaptants help to secure cargo molecules by trapping these receptors underneath the neck of the vesicle as it's budding. Each type of adaptant binds to different subsets of receptors, so cell membrane adaptants only bind to the receptors of proteins that belong in that cell membrane. This means adaptants are important for the sorting of the cargo. When a vesicle buds and gets released, it needs to go to the right place, fuse with the membrane, and give that organelle its contents. When the vesicle gets to its organelle, it needs to recognize the membrane and dock with it, and then it can fuse to the membrane. There are specialized proteins known as Rab proteins. These are clues on the surface of the vesicles, which gives ideas towards their origins and contents. Rab proteins are vesicle membrane proteins, which get recognized and bound through tethering proteins that are on the membrane of the target organelle. Each type of membrane has its own type of Rab protein, and because an interaction between these proteins and tethering proteins is needed for docking, it makes sure that the right vesicles are fusing with the right targets. Extra recognition happens from snare proteins, where interactions between these stabilize the docking process after Rabs and tethering proteins interact. Once this happens, membranes need to fuse to the vesicle to release the content, and the fusion also allows the vesicle membrane to become the target membrane. This is how membrane-bound proteins are delivered as well. Fusion can be delayed, where the vesicle docks can only be positioned for fusion when a cellular signal is given. Membrane fusion needs the vesicle and target membranes to be very close together, which occurs through the twisting of snares. This essentially winches the vesicle in, and fusion can then occur. The membranes are able to become one, and through exocytosis, the secretion of the proteins is able to happen. We're now on to our next topic, which is cytoskeletal fibers. These include intermediate filaments, microtubules, and actin filaments, which can also be called microfilaments. The first cytoskeletal fiber that we'll discuss is the intermediate filament. Because the main purpose of this filament is to provide tensile strength against mechanical stress on cells, structure really equals function. To allow the filament to be as strong as possible, it is formed in a rope-like manner. There's first a monomer, which is one single intermediate filament protein. This monomer wraps around another monomer to form a dimer. The dimers are able to come together to create a tetramer. Then tetramers bundle together, as we see in the image, and they create the rope-like structure that is the intermediate filament. Microtubules are made up of subunits called tubulin, which are dimers of two spherical proteins. The dimers stack together and interact with non-covalent forces to make part of the wall of the microtubule. There needs to be 13 protofilaments to make a single hollow microtubule. Each dimer has one alpha and one beta subunit, which alternate down the filament. This means microtubule protofilaments have a beginning, or the alpha, and an end, or the beta. Each protofilament faces the same direction, which gives the microtubule polarity. The ends are known as the positive and negative ends. Microtubules form and tubulin dimers are added to either end of the growing fiber, but more efficiently to the positive end. The polarity is important for microtubule function, and the two ends have to be different physically and chemically. Microtubules are the shipping lanes for cellular transport and have their directions delineated for transport purposes. They form rapidly and efficiently when the tubulin dimers are being added to a pre-existing microtubule fiber, but tubulins aren't able to form tubes from scratch. Centrosomes are the tubulin organizers, where they get tubulin to make the initial microtubule tubes, and more tubulin will be able to join more easily. This makes the short tube get even longer. Centrosomes basically need to get the ball rolling. They contain hundreds of ring-shaped structures that are made up of a third type of tubulin, and these rings serve as the nucleation site, which is where the microtubule formation starts. The negative end of the new microtubule is embedded in the centrosome, while the positive end points outwards towards the cell's edges. Growing microtubules show dynamic instability.
Once a microtubule has been started at the centrosome, its positive end grows out as the new dimers are added to it. Then the microtubule disintegrates without warning, shrinking backwards as the dimers fall off of the positive end. Sometimes it partially falls apart, while others it falls all the way back to the centrosome. This is known as dynamic instability. Each dimer is capable of binding to GTP or GDP. GTP binding dimers pack efficiently into microtubule fibers, while GDP binding dimers don't. When microtubules are being polymerized, dimers are added to the end of microtubule fibers much faster than the GTP that they are holding can be split. This is good because it results in a stable GTP cap. If polymerization slows too much, and the GTP in the tubulin dimers at the end of the microtubule is hydrolyzed before more dimers can be added, the end of the microtubule will fray, which can cause disassembly. The rest of the microtubule is also made of GDP binding dimer, so once disassembly starts, it can't stop. This goes from a frayed end to a whole frayed fiber. The freed tubulin dimers go back to the free dimers and wait to bind to GTP again. We're able to stop microtubules from falling apart by anchoring the positive end to something. If it's linked to something on both ends, the microtubule is stable and has to be actively disassembled. It is dynamic because the centrosome is constantly sending out new microtubules, where the ones that catch on to a target are stable and the ones that miss are retracted. Actin filaments are smaller and more flexible than microtubules, as well as more abundant. They don't usually exist on their own and typically bundle together in a cell. Just like microtubules, actin filaments can grow on either end, but it's easier and faster on the positive end. They can disassemble pretty easily if there are no associate proteins bound to them. Each free actin monomer is bound to ATP, and once it's incorporated into a filament, that ATP gets cleaved to ADP to reduce the strength of the binding for the monomer in the filament. This promotes depolymerization. Actin filaments need to be dynamic, so they need to be able to assemble and disassemble, no matter the cost of energy. There is a high concentration of free monomers, unlike microtubules. This is on purpose, where small proteins in the cytosol bind to the monomers and block them from joining the growing actin filaments. These proteins are responsible for regulating actin filament polymerization and keep actin monomers free and in reserve. If new actin filaments are needed, the proteins release the monomers, which are bound by other proteins to promote the assembly, and new filaments are made. Most actin-binding proteins, however, bind to already polymerized filaments. There are actin bundling proteins that hold actin filaments together in parallel bundles. There are also proteins that hold actin in the meshwork of the cell cortex, as well as proteins that can cut actin filaments into smaller pieces. This allows actin to become more fluid and gel-like.
Cell crawling depends on actin, where they sense environmental signals via G-protein coupled receptors, which we'll talk about in a little bit, and they migrate towards these signals using actin. This occurs in 3 steps. First, the cell pushes out protrusions, some sort of podia, at its leading edge. Then the protrusions adhere to the extracellular surface that the cell is crawling on. Lastly, the rest of the cell flows forward to the anchoring points, which fills in the cell at the advancing front. These steps are driven by actin in different ways. The first step of cell crawling is led by new actin polymerization. First, thin lamellipodia are pushed out, and they contain a dense meshwork of actin. This actin is set so most of the filaments have their plus end closer to the plasma membrane. The smaller protrusions are filopodia, which each contain a loose bundle of about 10 to 20 actin filaments, which is not a dense sheet, and their plus ends point outward. Both of these podias are very dynamic, so a crawling cell is always sending out these structures. It's the polymerization of actin in these structures and pushing the cell membrane out that allows for this to occur. The podias need to stick to a crawlable surface reversibly, and transmembrane proteins called integrins allow this. They adhere to molecules that are found in the cell's external environment, which anchors the cell to the surface it's crawling on. On the intracellular side of the membrane, the integrins are anchored to actin filaments, and they bridge actin to the surface that it's being crawled on. Before we go into the third step in actin, it's important to note that actin's not the only thing responsible for cell crawling. Everything in the cell is responding to extracellular clues, rearranging the cytoskeleton in response to the environment. The cues are signal molecules, and the cellular responses are mediated by membrane receptors. All cues that activate actin eventually culminate on a GTP binding protein, which is known as Rho. This is a small G protein. When activated, it can trigger actin polymerization and bundling to let the filopodia form, trigger the formation of lamellipodia, and trigger actin-myosin interactions to promote cell crawling. The integration of these signals from the outside environment and the cues from inside the cell can affect Rho, which ends up changing the shape and movement of the cell. The inside cues have info on the cell's size, health, nutritional status, and readiness for division. The activation of Rho triggers the actin and myosin needed for the cell to contract. All actin-binding motor proteins are part of the myosin protein family. Myosins bind to and cleave ATP, which gives energy of their movement along the actin filaments. Myosins go along actin from the negative to positive ends. Myosin and actin binding and movement drives the muscle contraction in our bodies. Myosin 2 is the major myosin protein type that's found in muscle, and myosin 1 is found in all cells. Myosin 1 has a head domain and a tail domain. The head domains bind to the actin and can bind or cleave to ATP. The tail determines what the myosin will move, but it differs between the myosin 1 proteins. Myosin almost acts like a human arm, where there are two planks connected by a joint, which moves when ATP is hydrolyzed. The lever arm protrudes out from the head domain of myosin when ATP is being hydrolyzed, and after this occurs and only ADP is bound, the lever rotates about 90 degrees, forming a bend. ATP hydrolysis is different. The binding of ATP greatly reduces myosin's affinity to actin, which allows the bind-pull-release mechanism of motion. Ultimately, it's the action between actin and myosin that allows for the third step to occur, where the cell pulls itself along to where the rest of the cell is anchored. This process is known as translocation, and it wouldn't be possible without actin and myosin being triggered by the activation of Rho. Alright, so now we'll move on to the topic of G protein coupled receptors. These are a type of surface receptors that carry signals into the cell through modified proteins. These usually occur as cascades, and they usually happen through phosphorylation. They use enzymes such as cyclic AMP and calcium as second messengers. G protein coupled receptors are synthesized at the rough ER using start and stop transfer sequences. They're shipped to the Golgi, then they're processed, and then shipped to the cell membrane using vesicles. As with any receptor, the first thing to happen to a G protein coupled receptor is that a signal molecule will bind to it, causing a shape change. When G protein coupled receptors change shape, it activates a G protein that's associated with the receptor found at the cytosol face of the membrane. All G proteins bind GTP and GDP, and when inactive, the alpha subunit of the G protein binds to GDP. If a signal molecule binds to the receptor, the change in the shape of the G protein decreases its affinity for GDP and increases its affinity for GTP. The G protein then lets go of GDP to bind to GTP. Those bound to GTP are active. Then, the alpha subunit can change target molecules, and the beta or gamma complex can do the same, creating a signaling cascade that flows into the cell. The G protein has GTPase activity, and when the G protein hydrolyzes GTP to GDP, it is bound to the inactive form, GDP. The subunits are able to reassociate and translocate back to the receptor in order to do the process again when a new signal arrives. The G protein basically turns itself off like a timer. G proteins activate and open membrane-bound enzymes and channels, and they affect enzymes at the cell membrane, having more cascading effects. The more common enzymes that are activated by G proteins are adenylate cyclase and phospholipase C, which are activated by different types of G protein coupled receptors. When adenylate cyclase is activated, it catalyzes the conversion of ATP into cyclic AMP, which is needed to carry the message into the cell. This means that cyclic AMP is our second messenger, and its presence alone helps to convey information. It normally activates a single protein, protein kinase A, which is important for signaling, and it achieves the physiological response of phosphorylation. Calcium acts as a second messenger in another G protein coupled receptor system. This also uses a membrane component called PIP2. It begins with a signal molecule that binds to a G protein coupled receptor on the outside of the cell. This activates the G protein, which then activates phospholipase C. This splits PIP2 into two other molecules. IP3 is another second messenger that goes to the cell's interior, and DAG stays in the membrane to activate protein kinase C. Protein kinase C phosphorylates many target proteins, such as calcium channels, which lets some calcium into the cell. IP3 goes to the endoplasmic reticulum to trigger the release of calcium from there, and calcium binds to PKC to make it more active, as well as calmodulin for the same reason. Calmodulin activates a different kinase to phosphorylate target proteins for other functions. Now we're on to our cell cycle. There are only two parts of this cycle that we can actually see, which are mitosis and cytokinesis. Collectively, these make up the M phase, which we'll discuss a bit more later. The rest of the cycle is collectively called interphase, which has three different subphases. The first is the G1 phase, which is the gap between the M phase and the S phase. There is then the S phase, which is where DNA synthesis occurs, and then the G2 phase, which is the gap between the S phase and the M phase. Throughout this cycle, there are three different checkpoints to ensure that the cycle is running smoothly. There's one in the G1 phase, which is the most important because it's the only one to get external cues. It checks the environment to make sure that it's good enough for the cell to go into the S phase and divide. If the conditions don't look good, the cell goes into the G0 phase forever, where it carries out all of its normal functions but does not divide. The second checkpoint is in the G2 phase to ensure that DNA replication and repair are complete. The last checkpoint is in the anaphase of mitosis, where it's made sure that the mitotic spindles are attached to the sister chromatids and have the proper amount of tension. There are key proteins that are responsible for the main events on each stage of the cell cycle, and they're turned on and off in a specific pattern to get the cellular effect. Cyclic regulation of proteins during the cell cycle is usually through phosphorylation and dephosphorylation. Kinases and phosphatases are regularly expressed but are regulated by cyclin and trigger the cell's progression through the cell cycle. Cyclins control the kinases, which control the proteins that are needed for the cell cycle. Cyclins bind to the kinases and activate them, making the kinases cyclin-dependent kinases. These can only be activated when its corresponding cyclin is made. The different types of cyclins are made for different phases of the cell cycle, so each cyclin-dependent kinase complex triggers different cellular events to prepare the cell for the next cell cycle stage. Cyclins get made, they bind to and activate cyclin-dependent kinases, and then they go off and phosphorylate target proteins which change the behavior of the cell. The cell cycle and stages get delayed partially by CDK inhibitor proteins, which block the interactions between cyclins and CDKs so that they can't be activated. If progression is possible or necessary, the inhibitor protein is removed. There are DNA damage checkpoints in the G1 and S phases from starting and completing the S phase if the DNA is damaged, and one in G2 prevents the M phase entry. Damage to DNA causes increase in expression of P53. This is a transcription factor that activates P21, which is a CDK inhibitor. This prevents the cell from going into the S phase so the damage can be repaired, but if it can't, the cell will undergo apoptosis through P53. P53 is a tumor suppressor gene, and if there are mutations to it, it often leads to cancer. So, without P53, P21 can't be made to respond to damage, and without P21, G1 or S CDKs aren't inhibited, so the damage is then replicated. Once the cell gets through the checkpoints, it's ready for the M phase, or mitosis and cytokinesis. This only lasts about an hour, and it's where the cell reorganizes everything in it, distributes everything equally, separates the chromosomes, and divides into two. This is all controlled by mCDK. mCDK forms when there's an increase in M cyclin levels, which begins at the beginning of the S phase and builds in the G2, then pairing up with its CDK. This is turned on by CDC25, and active mCDK activates more CDC25, which is positive feedback. In order to prep for chromosome separation, the centrosome needs to be copied so they can move to opposite poles, and then each one organizes the microtubules into mitotic spindles. This duplication starts at the beginning of the S phase and is triggered by S CDKs and G1S CDKs, both of which trigger replication. When mitosis begins, the centrosomes separate to form asters. During prophase, more microtubules are sent out from the centrosomes, and some microtubules from one centrosome touch the microtubules growing from the other centrosome. This stabilizes both of those microtubules, and these joined sets are the mitotic spindle. The centrosomes are spindle poles and anchor the spindles on opposite sides of the cell as it divides. Now all that needs to be done is the attaching of the sister chromatids to the microtubules so they can be pulled apart. Next is prometaphase, where the nuclear envelope breaks down due to phosphorylation. This lets the chromosomes be accessible to the microtubules. The microtubules attach to kinetochores, and because they face opposite directions, each chromatid gets linked to microtubules from opposite centrosomes. There is now tension on the chromatids and the microtubules, and the chromosomes get pulled back and forth, then end up centered down the middle axis of the cell, which is known as the metaphasic plate. This is what lets us know we're in metaphase. The M-phase checkpoint is this tension on the microtubules and chromatids, and if tension is not on all of them, the M-phase stops. If all of the chromatids are under tension, the cell completes metaphase and anaphase can begin. Anaphase starts by the release of the cohesion links that keep the sister chromatids connected. The sister chromatids can then be pulled apart. This is done by spindle microtubules bringing them to opposite poles, known as segregation, and separase chews up the cohesion proteins to separate them. Separase is activated by a bunch of proteins that also target MCDKs for destruction. This is the anaphase promoting complex. This means the beginning of the end of mitosis has been initiated. Anaphase is actually two parts. Anaphase A consists of the microtubules on the kinetochore shortening by depolymerization, which brings the chromosomes to each pole. Anaphase B is where the spindle poles get further apart, pulling the chromosomes even further. For anaphase A, motor proteins are attached to the microtubules that are adjacent to the kinetochore, using energy from ATP to disassemble the tubulin from the positive end of the spindle microtubules. Kinesin and dynein pull the centrosomes apart for anaphase B and may be associated with microtubules of the cell cortex. If they walk on these microtubules, the motor proteins can drag the spindle poles closer to the cell membranes to pull them further apart. Once segregation is done, the spindle microtubules disassemble, and telophase can begin by forming two new nuclei at the opposite poles. The vesicles of nuclear membrane from prophase cluster around the chromosomes and fuse to reform nuclei. The nuclear pores and lamins get dephosphorylated to reassemble. Cytokinesis then splits the cell to mark the end of the M phase and the cell cycle. This actually begins in anaphase. The contractile ring made of actin and myosin is needed for cytokinesis, and it constricts the cell in the center to pinch it off into two cells. Each daughter cell needs its organelles from the mother, and there are so many mitochondria and chloroplasts that it's pretty easy. The ER also gets cut in half by cytokinesis. The Golgi fragments into many vesicles during the M phase and then attach to the spindle microtubules through motor proteins, and they get rides to the opposite poles. Epithelial cells connect to things in 5 different ways. These are classified according to their functions, but altogether they're called cell junctions. There are tight junctions, adherence junctions, desmosomes, hemidesmosomes, and gap junctions. Tight junctions seal the cells together so there are no leaks. These are formed by claudins and occludins, which are made like a zipper on either cell, and when the proteins zip up, the cells get joined. In the case of this image, the claudins are the purple on the top, while the occludins are the blue structures on the bottom. Without these junctions, life wouldn't be possible because the molecules would leak between the cells instead of going through them. These proteins also help to define membrane domains. Adherence junctions, which are on the left of this image, and desmosomes on the right, bind one epithelial cell to the other through cadherin. This interacts tightly in neighboring cells. Cadherins get tethered to actin filaments for adherence, and in the case of desmosomes, they tether to keratin. This tethering is what allows the cell to be linked to the cytoskeleton. Hemidesmosomes bind an epithelial cell to the basal lamina. We see this in the top left portion of the image. In the blown up portion on the right, we see that the integrins in hemidesmosomes are linked to collagen in the basal lamina. Gap junctions are a more private form of communication between neighboring cells, and they are openings. Connexins meet between two cells to make a channel across the two cell membranes, and small inorganic and hydrophilic molecules can diffuse between cells through these. This makes the cells metabolically, chemically, and electrically coupled, and these junctions can be regulated in response to different environmental cues. Now we're on to our last topic, which is never an easy one. This is the topic of cancer. Cancer cells are mutated cells with abnormal behavior and breakdown. They break the rules for a good portion of biology. They should divide when new cells are needed, but not when they're not. They should undergo apoptosis when the need is past. They should stay where it needs to. They should maintain their cellular and biochemical characteristics. So, if a single cell breaks those rules, it's not really a huge deal. But if it goes through the cell cycle and divides, it can become a problem. It can create a mass of rapidly dividing cells that crowd, push, and destroy the tissues in the way. This is what cancer is. It's an expanding clone of abnormal cells. Cancer cells are defined by two properties that get passed on to their daughter cells. The first is that they proliferate way past the normal constraints, and the second is that they invade and colonize the other regions that are usually for other cell types. This is what makes them so devastating. If the cells have the first property but not the second, they create a benign tumor. This is shown in the left side of the image. Benign tumors remain localized and can usually be removed by surgery. A tumor is malignant if it has the ability to take over other areas, like we see in the right portion of the image. A metastatic tumor is where cells have migrated to and/or invaded a blood or lymph supply line, or the circulatory system. Then these cells can be taken to other parts of the body and create secondary tumors, which have the same properties as the primary tumor. Although there are environmental risks to cancer, it's basically just somatic mutations. There are always mutations that get through, but usually we're fine because there's random mutations. Cancer is caused by the accumulation of specific mutations that come together in a unique combination. Just to be proliferative and invasive, a cancer cell would need multiple mutations, and it's rare for the mutations to come together in that combination in one cell, unless a good amount of time has passed. This is why we normally refer to cancer as a disease of old age. Genetic instability is causally related to cancer progression, and when mutations affect the replication and maintenance of the genome, we see chromosomal mutations. This is the biggest cancer risk. These mutations may deactivate DNA repair enzymes, bypass cell checkpoints like p53, or cause issues in mitosis. They lead to more severe mutations as the whole genome can be changed. There are two main classes of gene mutations that could lead to cancer. The first are oncogenes, which become hyperactive when mutated, and the activity causes the cancer. In this case, the unmutated gene is a proto-oncogene. Genes that become inactive when mutated and cause cancer are called tumor suppressor genes. The protein normally prevents cancer, but the absence of it in the mutant causes the cancer. Oncogene mutations are genetically dominant, while tumor suppressor gene mutations are recessive. This recessive property explains the genetic predisposition that some have. If there are two healthy wild-type tumor suppressor gene alleles, you don't have cancer, but to have it, both of these alleles have to be mutated. This is the probability of mutating one allele squared. If you have one healthy wild-type tumor suppressor allele and one mutated allele, you don't have cancer, but to develop it, the one good allele needs to be mutated. This is the probability of mutating one single allele. This means that the risk for cancer is much higher than the other one, and is inherited, making it a genetic predisposition. For cancer cells, it doesn't matter if the oncogene has been mutated to be active or a tumor suppressor gene was mutated to be inactive. It only matters that the cancer cells are dividing. For most tumors, both of these genes are mutated. The more important thing is where the mutation is found and what gene it affects, what protein the gene encodes, and what pathway the protein is part of. Now, as much as we'd like it to be, it's not really possible to create a universal cure to cancer. This is because all cancer types have different mutations, and cancer cells mutate in real time. Ultimately, early detection is key, because the sooner the tumor is found, the smaller it is, the more easily you can remove it, and the less time the cells have had to mutate. If you use cancer's advantages against it, this seems to have success. For example, cancer divides rapidly, so DNA can't be repaired. Radiation, as shown in the image, allows the tumor to become damaged, and it can't be repaired. But the healthy cells are able to repair their own DNA. A different drug inhibits the repair pathway that cancer cells use, but it's pretty old, so healthy cells don't seem to use it. Other strategies include blocking new blood vessels from forming, which doesn't allow the tumor to get nutrients.
There are also vaccines that are being developed to allow your immune system to recognize features on the cancer cells and then fight it, like an infection. When we understand more about cancer, we're able to create treatments that target the cancer cells but leave the others unharmed. And with that, that concludes my eReport for Unit 2 of Cell and Molecular Biology. Thank you so much for watching, and if you have questions, please be sure to let me know.